So an enzyme is a protein responsible for speeding up essential biological processes in our body, which could otherwise take years to complete. When the genetic code instructing for the formation of an enzyme is being altered or mutated, this will result in the enzyme either not being produced at all or produced in a lower amount and will exhibit impaired or no activity. So in the case of ADA-SCID, it is commonly caused by a combination of different mutations in the ADA gene of the chromosome 20. So this is the chromosome 20 and this is the location of the ADA gene. This gene is then transcribed into a sequence known as the coding sequence or the mRNA sequence that is then translated into a protein. So two single nucleotide alterations on the same allele were identified from a patient prenatally, and this is highlighted in green and blue. So the first mutation is known as the Y97C mutation, and this is due to a substitution of the nucleotide A for a G at the nucleotide position 290 of the coding sequence. This is a zoom in of the coding or the mRNA sequence. And this leads to an amino acid tyrosine being replaced by a cysteine at the amino acid position 97 on the protein sequence, producing a mutated ADA protein sequence, and hence the name Y97C. The second mutation is the L106B mutation, and this is when a nucleotide C is being replaced by a G at the nucleotide acid position 316 of the coding sequence, leading to an amino acid leucine being replaced by a valine at the amino acid position 106 in the protein sequence. And then both of these mutations are caused by a single nucleotide change in the genetic code, and when both of them in the genetic code of the mRNA sequence, and when both of them are present together, this will completely abolish the activity of ADA enzyme. And this is attributable to the accumulation of toxic substances, including adenosine and deoxyadenosine, as the ADA enzyme is incapable of assisting their conversion.